Okay, this material is for Biology 160 and it's worksheet 6, and we are talking about cell structure and function. So that's what this short presentation is about, because I'm going to be gone for a couple of days. So here's the material. I'm just starting on page 24, and I'm going to do a couple of these items right off the worksheet. Go through those as if I was doing it more or less in class on the board. So we hope that the camera stays in focus and the technology works all right. But the first one says, below the diagram of a phospholipid, label the hydrophobic and hydrophilic regions, illustrate the organization of these molecules as they arrange themselves in the bilayer cells. So we have talked about phospholipids when we talked about the biological molecules. So this is representing the polar head of the phospholipid, and this is representing those two hydrocarbon tails of the phospholipid. So if you need to review that, go back and take a look at the biological molecules. So this part is the hydrophilic head that interacts well with water. It likes, put likes in quotes, likes water. Now that's what philia is, likes it, and it likes water. And these are hydrophobic tails. And phobias are fears, so this is the water-fearing end of the molecule. And they align themselves together to make up a membrane. So if we were to draw some more of those in together, they would arrange themselves sort of like this. Just to draw several of them, 4, maybe 5 in a row, it doesn't really matter. But they're all the same. We have the hydrophilic heads around. They have charged groups on them that interact well with the charged polar water molecules. And there's another row of them that forms, and their tails sort of point toward the other tails, sort of like yay, without getting carried away with the artwork. So you wind up having a bilayer. And this is referred to as either a lipid bilayer or a phospholipid bilayer. And what you have are, again, the hydrophilic heads, likes water, on both sides of the membrane. And these are all packed tightly together. They're not covalently bonded. They just interact and just stuff together with each other based on their interactions with water. And this region inside is a hydrophobic region. The water has literally been pushed out, because these tails, didn't mention it above, but if you remember from the earlier discussion, these hydrophobic tails are hydrocarbons. And you can go back and look at the structure, but it's just long chains of about, 18 or 20 carbons, something like that, on and on. And they are all saturated, or mostly saturated, with hydrogens. So the point is that these are non-polar regions of the molecule. They do not interact with water. Okay, so this is a water-free zone, perhaps you could call it that. But it's a hydrophobic region of the membrane. So the point is that anything that's dissolved in water, and water itself, cannot pass easily through this region. It's excluded. These are like oils, and oil and water don't mix. Okay, so that's the basic arrangement of phospholipid molecules, hydrophilic head, hydrophobic tails, and how they arrange themselves in a bilayer. Now, to continue this illustration, to make it a cell membrane, I'm not going to draw 8,000 of these little individual phospholipid molecules, but let's just pretend that we do, and we take this row, this layer, and it continues on and around, and we take this layer, and it continues on and around, and now pretty soon we have a cell, and this comes back up to the other side, and the same with the other one. And these two lines that I'm drawing are representing the phospholipid bilayer of the cell membrane. Phospholipid bilayer. And we typically just call this the cell membrane. Now this gets to be confusing, or not confusing necessarily, but time-consuming to draw a cell membrane with both of these phospholipid layers making up the bilayer. So typically what we draw for a cell, understanding that this is what makes the membrane up, we typically just draw a cell like that. And there is our cell. And we understand and know that this phospholipid bilayer is what's represented by that single line. That just saves us the time of having to draw the double line or even drawing all those phospholipid molecules. You know that that's what makes up this cell membrane. And if we label it the cell membrane, we're good to go, and we can move along and cover more material quicker. So, I want to show you a picture that's out of your book on this cell membrane. So, here is what's in your book, and you may have to magnify this. If I try to magnify it, I think the camera freaks out and can't get it in focus. But what you see if you look at it carefully, and you may need to get a magnifying glass, I sometimes do that, these little heads that look like little pink golf balls are all of the phospholipid heads. And here are the little tails, and there's one layer, and then there's another layer. And this is all very flexible and fluid. These are basically little oil molecules. And that makes up this phospholipid bilayer. And that's what I was trying to draw on the paper. Now, another thing that this picture shows that we'll be talking about shortly is that the membrane is not just phospholipid molecules. A living cell membrane has all sorts of other things stuck in it, mostly proteins and carbohydrates and sugar. So keep that in mind as we go through this discussion shortly on transferring things from one side of the membrane to the other, transport mechanisms. Because we're going to have to put in little channels to let things flow through. Because remember, water cannot flow through here on its own. It's excluded by those hydrophobic tails of the lipid membrane. So if water is going to pass from one side of the membrane to the other, it has to go through little channels. And other ions, nutrients that are dissolved in water, the only way they can move through is to go through a channel also. And water and the sodium or water and the glucose are able to move through and pass through that hydrophobic region. And there are lots and lots of different types of channels. Some are pumps, some have gates on them. There's all sorts of protein that gets associated with the cell membrane. And sometimes there are little sugar groups that are attached to a lipid. So we have glyco for sugar plus lipid. So these are sugars that are attached to a particular lipid. We can also have glycoproteins. Glyco is referring to sugar, glycogen, and the protein. So here are individual little sugar monomers, sort of a branching chain of sugars that are attached to a protein. So this is then referred to as a glycoprotein. That's pretty straightforward. There are other molecules that are in the membrane also, such as cholesterol. You remember what that looks like. It was a series of 4 rings, a type of lipid. It's embedded in the membrane and it gives rigidity and sort of some structure to the membrane, sort of helps solidify it. And what else do we have in here? Not shown well are certain proteins. All the proteins appear to be blue. Some of them sort of wrap through here several times, and then they can have functions on one side or the other. Peripheral proteins are shown on one side of the membrane or the other, and then there are integral membrane proteins that go all the way through the membrane, such as this one. This figure also shows the cytoskeleton, filaments of cytoskeleton, which are these. It's all protein. And there are different types of protein. This would be inside the cell, and this would be outside. So these intracellular proteins often hook up with peripheral proteins on the cell, and that's what keeps the cells anchored in place so they don't drift off. Your skin cells don't migrate into your stomach and vice versa. They stay where they're supposed to, anchored in place by this cytoskeleton. Let me back up a minute. Sort of talk myself into a corner. This cytoskeleton is inside the cell. So forget about keeping the cell in place. That would be from the anchored from the outside. The cytoskeleton gives it its shape. So for example, a red blood cell in cross-section looks sort of something like this. It sort of looks like a flattened out donut. That's in cross-section. If you were to look at it from a top view, it would look sort of something like this. That's supposed to be round, and it would have sort of a squished little donut region in it. It looks like an inner tube almost that hasn't been punched through. That shape is dependent or determined by the cytoskeleton. But that's getting ahead a little bit of what I wanted to talk about. I wanted to show this figure to discuss the makeup of the phospholipid bilayer with the phospholipids and other associated proteins, cholesterol, other lipids, and other sugar groups. Okay, so let's go back to the worksheet packet. It talks about what are the main functions of the cell membrane. Well, one of the main functions of the membrane is to keep inside what's supposed to stay inside and to keep out what shouldn't be out. So when we get to transport discussion, we'll see that there is a very high concentration of sodium outside the cell. So I'll just indicate that with an up arrow for elevated, and using standard notation I'll just put this in square brackets, and the brackets just translate to concentration of whatever inside the bracket. So this reads, if you were to look at this part of the figure, you would understand this to mean that there's an elevated concentration of sodium out here in this extracellular fluid. An extracellular fluid is fluid that's outside the cell. That's what the extra means. It's outside the cell. And since we don't like to abbreviate extracellular fluid, we don't like to spell that out rather every time, we can often just use ECF for extracellular fluid, and I'll use that abbreviation extensively. Okay. Without looking at how this concentration becomes high on the outside right now, we can note that it's just the opposite inside. It has a low concentration of sodium inside the cell. And this is in the cytosol, or inside the cytoplasm. And by the way, since I just mentioned the two terms, you should be clear on the difference between cytosol and cytoplasm. Look it up, write down the definition. If it doesn't make sense, bring that question to class and we can talk about it quickly and clear that up. Okay, so just looking at sodium, and we're talking about functions of the cell membrane, one of the functions is to keep the sodium from just passively diffusing through the cell. It can't on its own because the sodium is attached to the water. We've already talked about ions and how they interact with water. So the membrane separates and prevents movement across the membrane sort of randomly or passively. If sodium is going to go across the membrane, it cannot go through this phospholipid bilayer without a channel. So we would put a channel, or show a channel. So there is a channel. This is a protein, just like in that illustration that I just showed you. And I'm just representing it as a tube with a hole through it, so that the sodium, or anything else, depending on the channel, can then flow into the cell. And it would enter that way. Now we'll go through more of these details in shortly, but this is the basic idea of what has to happen. If it's going to be in the water, which sodium and all other ions are, and it crosses the membrane, it has to do it through some sort of protein channel or pore or gate. Another function of the membrane is that there are, again, lots of different proteins in the cell membrane. So some cells, pretty much actually all cells, have protein receptors. And these are proteins. Remember, this is a phospholipid bilayer, and some of these proteins are woven through the membrane, and they kind of project to the extracellular side. This is outside the cell, and they act as a receptor. These are chemical receptors, and they bind chemical signal molecules. Signal molecules. There's a lot of information coming at you fairly quick, but it's the material that we're going to be using for the next several courses. So introducing it now, I think, helps. This is protein. So don't forget that. And it works something like this without going into any details. Here comes a signal molecule. Let's pretend. Maybe it has come through the blood. And this signal molecule, I'll just abbreviate sigmol for sig molecule. It could be a hormone. So e.g., for example, a hormone. And in A and P, we'll look at the endocrine system that discusses hormones. So at any rate, back to the receptor, what happens is that you have specific receptors for specific signal molecules. The molecule binds to the receptor on the outside of the cell. And what that does is it triggers a series of events inside the cell. Sort of a sequence of things that happen. And we're not doing the details of this now, we will soon. But that's just another function of the cell membrane, as it acts for a way of the cells to communicate with each other through either hormones or other mechanisms. Okay, let's see if that works. This should give you plenty of questions to talk about. Number 3 on the worksheet is proteins embedded in the plasma membrane have many different functions. Explain several major functions of membrane proteins. I think I've already done some of that. Receptors, channels, pores, integral membrane proteins from the illustration in the book, etc. Okay, the next couple of problems in the worksheet packet deal with prokaryotic, eukaryotic cells. You can draw that and color it and do that on your own. I'm not going to spend time in class. I've talked about the differences enough that I wanted to talk about. The basic structure of a prokaryotic cell versus a eukaryotic cell, again, you can do that on your own. And if you have questions, please bring those to class. So the next thing that I'm going to talk about, I'm going to take a break here shortly, turn this off, and then start another one. But we will spend some time going through page 26 of your worksheet packet, looking at the different organelles and the various functions of those inside the cell. So I would encourage you strongly to read that section in the book on the different organelles. And they're listed in the book more or less, I don't know if they're in that order or not, but we'll talk about the nucleus, the nuclear envelope, etc. going on down through that list. But please look at that ahead of time. It'll make more sense when we talk about them here in this recorded lecture.